Hello everyone, welcome to this session regarding bacteria. Today we'll discuss the cell structure, components, the types, positive and negative, shapes, spores, and antibiotic related topics. So beginning with what are bacteria? Bacteria, as you all must be knowing, they are prokaryotic structures, basically prokaryotic, unicellular structures. Okay, so what are prokaryotic? Let's break it down. Prokaryotic means basically means primitive. Okay, so basically these are cells which have primitive nucleus. Now the question is, do they really have a nucleus? Now this is the first question that I'd like to ask to you all. So do they really have a nucleus? No. Okay, so they don't have a nucleus. What they have is, I'm gonna show it to you right now. So for example, if this is a cell, this we can say cell wall, inside of which there will be a cell membrane, and here this is a kind of structure that they have. So is it a nucleus? No. What is it then? It's a chromatin. Okay, so what they have is a chromatin which is circular, as you all can see, and it is single stranded. Okay, chromatin which is circular. Now they don't have a nuclear membrane. Okay, they don't have a nuclear membrane. Normally what our cells have is, if this is cell wall, we have a nucleus which has nuclear membrane, nucleolus, and chromatin, right? This is the chromatin or DNA, and this is a nuclear space which is nucleolus, and this is the nuclear membrane. So together we call it a nucleus. But in case of bacteria, they don't have it. All they have is a chromatin material which is circular in arrangement. Okay, so will we call it a chromatin? No. Will we call it a nucleus? No. So what we call it is basically a nucleoid. So nucleoid is nothing but amorphous density containing chromatin. Okay, so chromatin is basically the nucleoid. Nucleoid basically is amorphous collection of circular DNA in case of circular DNA, or chromatin in case of prokaryotes. The first thing is clear. Now I showed you a cell wall, a cell membrane, and a chromatin. What else? So they have ribosomes. Okay, for example, this ribosome, and this ribosome is 70S. So mind you, humans have 80S. Okay, humans have 80S. That is, eukaryotes have 80S, and prokaryotes have 70S ribosomes, which is clear now. Now, who all fall under this category of eukaryotes? Humans for sure. Who else? Fungi. Okay, for now you can consider fungi as eukaryotes. With subsequent chapters we'll learn more about fungi and other eukaryotes, but for now, one example of eukaryote is human, the other one is fungi. And prokaryotes basically includes bacteria. Now moving forward, so till now we have discussed that they have a cell wall, cell membrane, ribosomes, 70S, and a nucleoid, right? And now talking about the cell wall in detail. Okay, so cell wall basically has peptidoglycan in case of bacteria, and mind you, chitin in case of fungi. Okay, now cell wall, what is it made up of? Like cell wall basically are of, like, depending on the cell wall we can classify bacteria into two types: gram positive, gram negative. Now why this difference? Now let's break it down. For example, if this is the cell membrane, okay, let's make it this way. If this is cell membrane I'm making it bilayer. Why? You know it, fluid mosaic model, bilayer. So if this is the cell membrane, okay, this is the cell membrane, this is the peptidoglycan layer in case of gram positive, and in case of gram negative, this is the peptidoglycan layer. So you can see the difference. Peptidoglycan layer is very thick and is very thin in case of gram negative. Now I've made one space here. If you've noticed, the space here, what is it for? So basically, gram positive, they just have one cell membrane, right, just like the negative. But gram negative, in addition to the cell membrane or plasma membrane, they have one outer membrane too. So they have an additional outer membrane, okay, similar to this plasma membrane, they have an outer membrane, and between this actual cell membrane and an outer membrane, the space which lies between it is known as the periplasmic space. Periplasmic space, okay. This periplasmic space is the space between the outer as well as the inner membrane. Now, what is this inner membrane? This plasma membrane layer, in case of gram negative, is known as inner membrane, while in case of gram positive, we may still call it cell membrane or plasma membrane. So cell membrane, plasma membrane, in case of gram negative, is known as inner membrane, because it, in addition to this inner membrane, it also has an outer membrane. So simplify it, this is the cell membrane in case of both gram positive and negative, and this cell membrane, and in addition to this cell membrane, gram negative, it also has an outer membrane, okay, and this space between this outer membrane and the inner membrane is the periplasmic space, and this, suppose, is the peptidoglycan layer, okay, and in case of gram negative, this is the peptidoglycan layer. So you can see the difference, and so let me jot it down for you. So cell membrane, present, which is also known as inner membrane, okay, then no periplasmic space. In case of gram negative, there is periplasmic space present, okay. The peptidoglycan layer is thick, is thin, and here it is thick. Now what else? Outer membrane present, outer membrane absent, okay. Now in addition to this, they both can have, this is a flagella and pili, all right. But in case of gram positive, there is something else that they have, and that is teichoic acid, okay. So let me make it simpler for you. This, my friend, is flagella, this is pili, which both share, and this is teichoic acid, which is present only in gram positive, okay. So teichoic acid is present, teichoic acid is absent. What else? Pili and flagella can be present in both, but mind you, pili is seen mostly in gram negative. So mark it down, pili is seen mostly in gram negative. Now, so this is the comparison that I wanted to make. Let me just tell it again. So cell membrane is present in both the cases. In negative, we call it inner membrane. No periplasmic space in gram positive, but there is periplasmic space in case of gram negative, and in inside the periplasmic space is the peptidoglycan layer, which is very thin in case of gram negative and is very thick in case of gram positive. Now what I'd like to add here is whatever you see outside this inner membrane or the plasma membrane, all these things come under the cell wall. Clear? So whatever you see outside this inner membrane or the plasma membrane is the cell wall. So basically in case of gram positive, cell wall is basically the peptidoglycan layer. Clear? Peptidoglycan layer that we have. And you can also add there are flagella, pili and teichoic acid. These three structures are present in case of gram positive's cell wall. But in case of gram negative, what do you see? This is the normal inner membrane. Okay, so what does gram negative's cell wall consist of? It consists basically of the outer membrane, number 1, the periplasmic space, number 2, peptidoglycan layer, number 3, flagella 4, pili 5. Got it? So 5 things are present in the cell wall of gram negative. But in case of gram positive, it was the peptidoglycan layer number 1, teichoic acid number 2, and additional to it there can be pili and flagella. Pili, as I said already, is seen in gram negative mostly. Okay, now we saw that this is this is bacteria, okay, which has cell wall, cell membrane, which is also called inner membrane in case of gram negative. Then we saw one circular chromatin, right, which is known as nucleoid. And along with it we saw ribosomes, which were 70s. Okay, now diving deep into the cell wall again, we said gram positive, gram negative. Gram positive basically had peptidoglycan layer and teichoic acid, whereas gram negative had peptidoglycan, outer membrane, periplasmic space and pili, flagella in both the cases. Okay, now talking about the peptidoglycan layer a bit more in detail. Peptidoglycan layer is basically made up of NAM-NAG arrangement. Okay, these are nothing but sugars in peptidoglycan itself says it is made up of sugar. So the arrangement is like NAM-NAG, 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 N-acetylmuramic acid, N-acetylglutamic acid. Okay, so NAM-NAG arrangement is present throughout. Okay, now suppose and in this muramic acid chain there is one side chain consisting of tetramers, tetrapeptides, that is 4 peptides, A, G, L, A. Okay, A stands for alanine, G for glycine, L for lysine, and A again for alanine. Okay, AGLA. And here also in NAM you will find AGLA. Now this L and this A are connected by a tetrapeptide. Okay, 1, 2, 3, 4, 5 tetrapeptide cross link. Okay, so AGLA of one side chain is connected to AGLA of another side chain via a cross link. Okay, as simple as that. So in case of gram positive, this cross link is a tetrapeptide cross link, which is done by transpeptidase enzyme. Got it? By transpeptidase enzyme. But in case of gram negative, mind you, AGLA can be connected directly. Direct connection or crosslink. No tetrapeptide. Okay, so this is a very big difference seen at the molecular level. Now, summarizing everything, gram positive, gram negative, you know the basic difference that peptidoglycan is very thick here, very thick here, it is thin here, it has outer membrane and periplasmic space. Here, no such things, and here teichoic acid is present, here no teichoic acid. Okay, this is the basic difference that you had. Now talking about the peptidoglycan layer, which is in molecular level. Okay, this difference is at the molecular level. Peptidoglycan, here AGLA arrangements, they are connected by transpeptide, okay, side chain. Here there is one direct connection. Okay, now you'll think, why is there a direct connection? Now the answer to that is this L, lysine, is replaced by DAP in case of negative. Okay, because this lysine is replaced by DAP, that is diamino pimelic acid, there is a possibility of direct connection between AGLA's L and AGLA. Why? Because this L is replaced by DAP. So in case of L, there is DAP, DAP. So DAP and alanine. Okay, DAP of one side chain forms a cross link with alanine of another side chain directly. Okay, so because of this agla cross link, there is stability to the cell wall. Now I'll show you how. For example, if NAM NAG arrangement was like this only, NAM NAG arrangement was like this only, the structure would be like this, right? But since there is one cross link possible, it provides stability to the overall structure. Now, if one cell wall was just arranged like this, would it be stable, or would cell wall which is arranged like this be more stable? Obviously this, because cross links provide stability. Now going back, let's see how much have we covered up. So cell is done, structure is done, positive and negative are done, components are done to some extent. Now I'll dive into the essential and non-essential components, and we'll talk about the shapes, spores and antibiotics. Okay, now before I dive into the components, let me first discuss this scary structure. What do you think this is? Let's go down. This thing is nucleoid. Simple. This outside thing is the cytoplasm. Okay, now do you see these dots? These are ribosomes. figured out. Now, what do you think this green thing is? Okay, what do you think these structures are? Villi. This long structure is flagella. Got it? So it's a very easy diagram now. Now again, this is even simpler. Okay, this coiled thing is chromosomal DNA or nucleoid. This is ribosome. Okay, additional to that, you have cell membrane, cell wall. This is the cell membrane, this is the cell wall, and in some cases you'll even find capsule. We'll talk about it later. And this is the cytoplasm. If you can see, long structures are the flagella and small hair-like structures are the villi or fimbriae. Same thing. Now we'll dive into a very simple diagram of bacterial cell. Now you tell me, this thing is DNA or nucleoid. This is cytoplasm, this is ribosomes, these are plasmids, we'll talk about them, and these are flagellas, and if I had made something like this would be villi. Okay, so we covered up a lot. Now let's go into the structures. So cell wall is done in detail. Okay, cell wall is made up of peptidoglycan. Done. Cell membrane, done. Now I'd like to add something in the cell membrane. So normally cell membrane, we imagine it to be like this, but in case of bacteria, cell membranes are invaginated. This invagination is known as mesosome. Clear? Now what is this mesosome used for? Mesosome, why? Oh, you just imagine, why would this structure be needed at all? Obviously for transport. Okay, now we looked into the cell wall, we looked into the membrane. Okay, now we'll even looked into mesosomes, we looked into ribosomes. Okay, now I like to add something. This is cell wall, this is mesosome, cell membrane, chromatin is sorted, ribosomes are sorted, but is there any possibility of extra chromosomal circular DNA? Yes, extra chromosomal circular DNAs are known as plasmids. Okay, so in addition to that, you might as well find plasmids. Okay, you won't find other organelles. Other organelles like mitochondria is absent. Okay, so mitochondria is absent in case of bacterial cell wall, but you may find plasmids. What is the role of plasmids? Very simple. Extra chromosomal DNA, right? So they are responsible for the synthesis of pili. Okay, now this pili thing has come up a lot. You know what pili are? These things. You know what they are used for? They are used for sexual, or I mean, as well say conjugation. Okay, conjugation, that is meeting in a way. But remember, bacteria don't undergo sexual reproduction. So what are these pili used for? These pili, imagine it to be like this. Okay, this is one bacterial, it's another bacteria. These pili basically transfer materials. Okay, they are also known as sex pilus. Got it? So this plasmids in the bacterial cell, these plasmids synthesize pili, number one. Number two, they form enzymes such as beta lactamase or cell wall resistant cell wall inhibitors, or even you can say, you can call it like enzymes or structures which help in providing antibiotic resistance, or even like repair genes, DNA repair genes and all are synthesized. Okay, so pili and other enzymes which are responsible for providing resistance against the antibiotics. Okay, now we discussed about the plasmid also. I'd like to say something else now. This is the cell wall. This is the cell membrane. So in case of some bacteria, there might as well be structures outside the cell wall, and this is nothing but the capsule. Okay, capsule. So why is there the need of capsule? But like before starting that also, I'd like to say, if this is cell wall, this is cell wall, some have capsule. This is capsule. Okay, in some there are just amorphous densities. Okay, this is capsule, and some have amorphous densities. Now what is this amorphous density and what is this capsule? Okay, this amorphous density is glycocalyx, and this capsule is the one which provides it some protection. Now protection, how? Protection, basically if the cell has one capsule to itself, then it prevents phagocytosis. How? Because the antibodies cannot bind to the cell wall. Okay, cell wall is basically antigen, and these antibodies cannot bind to the antigen. They cannot bind to the cell wall because of the capsule. So they prevent immune stimulation, no immune stimulation, and because there is no immune stimulation, there will also be no phagocytosis, because the phagocytes or the cells like macrophages cannot phagocytose a capsulated bacteria. Okay, so two things, anti-phagocytic, and the other one is no immune stimulation. So capsules are for protection of the bacteria. Okay, and glycocalyx. So what is the difference between capsule and glycocalyx? Basically, capsules are structures which have a fixed structure to itself. Okay, but this glycocalyx, they don't have a fixed structure. They are just like amorphous densities. Now this glycocalyx basically helps in adherence. Mind you, this capsule also helps in adherence. Okay, obviously it's sticky, so it helps in adherence, but more to that, capsule is also important for protection of the bacteria, that is for antiphagocytic and anti-immune responses. Okay, but glycocalyx on the other hand is just for adherence, and it helps to cause long-term infections and is a virulence factor. Now I'll show you the example. Suppose a person, he has a prosthetic valve. Okay, prosthetic heart valve. Now if the bacteria has glycocalyx, then it will be very sticky and it will stick to the heart valve. Okay, if it sticks to the heart valve, what happens is it will cause a long-term infection. Okay, and this is responsible for its virulence. It won't be removed easily. It will stick to the heart valve. Okay, and the example of this, a very famous organism is Staphylococcus epidermidis. First bacteria that I like mentioned in this bacteria in this session is Staphylococcus epidermidis, which has glycocalyx. Okay, now what are the bacteria having capsules? So for now, just remember all the bacterias, okay, which go to the chest or to the brain. To the chest or to the brain have capsule because they need protection. Okay, now I'll mention like very important ones, but like for now, just I'll write the names, but for now, just like you can ignore it. In the subsequent sessions, you'll understand why and how. One example is Streptococcus pyogenes, Streptococcus pneumoniae. Okay, Streptococcus pyogenes, Streptococcus pneumoniae, Neisseria meningitidis, Neisseria meningitidis, Haemophilus influenzae, Okay. For now, like, I'll just mention 4 names. 1, 2, 3, and 4. Okay. There are a lot more, but we'll come to it later. For now, just remember Streptococcus pyogenes. Okay. This pyogenes is found in the throat. So it will obviously have a capsule. Streptococcus pneumoniae. Pneumonia related to chest. It also has capsule. Haemophilus influenzae related to chest. It has capsule. And related to brain, Neisseria meningitidis. It also has a capsule. So 4 examples for now. Now we completed cell wall, cell membrane, capsule, pili, even flagella. Flagella are basically nothing but organs for locomotion. And mind you, all these things, cell wall, capsule, flagella, they are antigenic. So in any antigen antibody test will be positive in case of these organisms. Okay, so like they are like extra things through which we can identify the bacteria. So in lab also, because of this flagella, which has flagellin protein, we can easily identify it. Okay, so we completed the cell wall, flagella, pili, chromatin, ribosomes, and even plasmids. Now, just so you may be interested about pili. Okay, these are pili. Pileated organisms. Pileated organisms are basically gram-negative organism. One example is E. coli. If you want to remember for now, E. coli. Okay, all the gram-negative bacteria's example E. coli. Flagella, this thing, it can be of 4 types. One, it is like this. Okay, peritrichus. That is all around its periphery. E. coli is an example. Another can be like this also, Lophotrichus. Just one. Amphitrichus, 2 sides. And Monotrichus. Okay, so the one just having one flagella is Monotrichus. Having a tuft of flagella at one end is Lophotrichus. And having one at each end is Amphitrichus. Amphi means both. Okay, so example of Monotrichus, very famous example, Vibrio cholerae. Okay, Lophotrichus can be Helicobacter. Okay, Helicobacter pylori. So imagine one helicopter, right? Helicopter is Lophotrichus, right? You can imagine one helicopter. And Amphitrichus, easy to remember, snake or spirillum. Okay, so if you imagine the structure and one flagella at each end, it is spirillum. So examples of all 3 is done, and peritrichus is E. coli or gram-negative Enterobacteriaceae. For now, just remember E. coli. So this is also summed up.